Hello students, and welcome to the second block of lectures of this module. In the last lecture, you heard about the basic mechanisms of transcription, RNA polymerases, the general or basic transcription factors, and the different elements of the promoter of a gene. Today we will focus on other proteins and DNA segments that together regulate the activity of RNA polymerase II and the rates of transcription of protein-coding genes. After this lecture, I would expect you to be able to describe the molecular basis of eukaryotic transcriptional regulation, its organization, its elements, either DNA or protein, and how they operate. Also, you should become familiar with some experimental methods used to define and characterize these regulatory elements. I also hope that with this lecture, you will also be aware of the many and amazing biological phenomena that are underpinned by the regulation of transcription. For instance, the process of embryonic development requires the constant, progressive narrowing down of the final differentiation choice of the embryonic cells. This requires the continuous re-evaluation of which genes should be turned on or off to subdivide the embryo into body structures of increasing complexity and detail. This requires regulation of transcription with exquisite spatial resolution. In addition to space, transcription rates need to adapt to time. The obvious example of this is our circadian clock, which ensures that certain genes will be more actively transcribed at day than at night, and vice versa. This ensures that the body uses certain gene functions when they are more needed. Sometimes it is not the spatial or the temporal precision that is needed, but to adequately regulate the amounts of protein. This is important, for instance, for heat shock proteins. These are needed in all cells all the time, but at much higher concentration when there is heat stress. So their transcription is quickly elevated precisely when temperature rises, and is reduced when the temperature is more normal. Regulating protein abundance is important not only for individual proteins but also in complexes whose function will depend on the stoichiometry of their components. For instance, complexes of the BCL2 family regulate the mitochondrial pathway of apoptosis, with some family members being pro-apoptotic and some others being anti-apoptotic. The appropriate activation of apoptosis, and therefore the fitness of the organism, will largely depend on the capacity to regulate the transcription of these proteins. This has practical importance, as this piece of news suggests. The internal clock of the body is likely to make the heart more resistant to surgery during the afternoon. Gene expression controls how a gene directs the production of protein, but the whole process of gene expression comprises several steps. You will remember that as transcription occurs, the resulting RNA molecule, the primary transcript, is processed by the splicing machinery into a mature messenger RNA, which is later exported from the nucleus into the cytoplasm. In the cytoplasm, the messenger RNA is bound by ribosomes which translate it into protein. So we perhaps should be not paying so much attention to transcription and rather to all these other processes, which are closer to the production of protein. The answer is not entirely. Thanks to modern high-throughput methods such as RNA-seq, tandem mass spectrometry, and others, we know that there is a good correlation between the rates of transcription of most genes and the abundance of the protein they code for, so that roughly 73% of the abundance of a protein can be determined just on the basis of how transcription occurs for that gene. This correlation, of course, is not perfect, and for some individual genes the rates of transcription may not be very informative of the amount of protein, but as a general rule, transcription is the best first approximation to the expression of a gene. Should we care about this as human? Is this relevant in medicine, for instance? Well, let us think about two basic consequences of regulating transcription. As we've just said, it allows to control the abundance of protein. This will include proteins that regulate, for instance, cell proliferation, such as the oncogene MYC, or immune cell activation, for instance, pro-inflammatory cytokines. So failure to appropriately regulate these types of genes can contribute to diseases like cancer or autoimmune diseases. Transcriptional regulation also allows the differentiation of cells, which often involve becoming postmitotic or with limited proliferative potential. So failure to implement or sustain the state differentiation can lead to the differentiation and reacquisition of proliferative capacity of some cells, which again can contribute to cancer. Also, during aging, the differentiation programs are not maintained so strictly, which leads to the functional decay of the cells and the tissues they reside in. Just as an example, consider the oncogene MYC. MYC is a transcription factor, a protein dedicated to regulate the transcription of many other genes. And this includes many proteins that regulate cell proliferation and survival. If you look at the type of mutations that are found in MYC across all cancers, by far the most abundant change is the amplification of the number of copies of the gene in the cell. This simply means that there will be more DNA molecules from where to direct the transcription of MYC, and therefore more MYC protein. You do not need an activating mutation in the protein sequence, like in other oncogenes like RAS or RAF. You just need to increase the amount of protein for MYC to become a problem. As MYC is also a transcriptional regulator, this will impact the transcriptional rates of many other genes. So this goes to show how important it is to regulate transcription. This is also illustrated by congenital defects in embryonic development. As transcription factors orchestrate the activity of many genes, they are key to induce the formation of new cell types or body structures, and their mutation can suppress the formation of these parts or induce the formation of additional ones. This is the case of polydactyly, which can be produced by a mutation in the homeotic gene HOXD13, which encodes a transcription factor of the homeobox family. The mutation of another homeotic gene, ultrabithorax, UBX, does not lead to suppression or induction of structures, but to transformation, in this case of the haltere, a balancing appendage in the thorax, into a wing. Other transformations can be seen in plants, with this triple mutant having a flower where petals have become sepals.